Hey, it's Scarlet Star here. I'm going to present the topic DNA in front of you guys, and it's DNA, and that is deoxyribonucleic acid. So, my reference is Molecular Biology of the Cell, 6th edition, and it's Bruce Albert's book. So, basically, this is the DNA which exists in double helical structure. This is the double helix form, and this is base pairing rule, means adenine binds to T and G binds to C. Adenine pairs with thymine and cytosine combines with guanine. But in case of RNA, adenine pairs with uracil and cytosine combines with guanine. There is no role of thymine in RNA. And this is parental DNA double helix, and this is template strand, and it is new strand. And this is template strand. Template strand was from here. That was parental DNA double helix. And now we are going to basically discuss what is DNA. So all cells store their hereditary information in the same linear chemical code DNA. Means all cells store their information in the DNA chemical codes that are nucleotides, which are going to pass to our cells like offspring. Like let's say if parents are containing that type of information, it is going to pass to offspring. So that's how it happens. All living cells on earth store their hereditary information in the form of molecules of DNA. Means they contain their hereditary information in the form of DNA. Like what mother and father have the characters, they are going to pass down to offspring or daughter cells. All cells replicate their hereditary information, and they are going to replicate their hereditary information. Like it happens in bacteria also. The mechanism that make life possible depend on the structure of the double-stranded DNA molecule. Each monomer in a single DNA strand, that is nucleotide, consists of two parts. A sugar deoxyribose with a phosphate group attached to it, and a base which may be either adenine, guanine, cytosine or thymine. So we can confirm it from here. Means if we have taken this backbone of the DNA, it's going to contain sugar phosphate backbone, and the inner part of the DNA is going to contain A nucleotides, AGCT content. Like we can see, A bonds to T and C bonds to G. It's a triple bond when G bonds to C, and it's a double bond when T bonds to A. And this is the sugar phosphate bond, which is mentioned in the diagram. And this is double stranded DNA. Hydrogen bonded base pairs. So next we can conclude that A sugar is linked to a next via the phosphate group, creating a polymer chain composed of repeated sugar phosphate backbone with a series of bases. The DNA polymer is extended by adding monomer at one end. Means this sugar phosphate backbone contains repetitive sugar phosphate backbone. Means this is a repetitive sugar phosphate backbone. This is the backbone. It is showing in the linear in the above figure. It is sugar phosphate repetitive backbone. So, and nitrogenous bases are inside of the DNA structure. DNA and its building blocks. DNA is made from simple subunits called nucleotides, each consisting of a sugar phosphate molecule with a nitrogen containing side group or base attached to it. The bases are of four types: adenine, guanine, cytosine, and thymine, corresponding to four distinct nucleotides labeled A, G, C, and T. So it's contain. So DNA is made from simple subunits. Subunits of DNA is nucleotide. Nucleotide is a subunit of DNA, means sugar, phosphate, and nitrogenous bases, obviously, and we can label it ATGC. ATGC. Adenine pairs with thymine, which is a here double bond, like here we have shown here. It's a double bond, and in the above figure, and this is a triple hydrogen bond. This is triple hydrogen bond which G pairs with C, guanine pairs with cytosine. So this is shown in here in this figure, in the below figure. G pairs with C. So it's a double helical structure, and so introduction of, basically what is the introduction of DNA? DNA and RNA molecules are linear polymers built from individual units called nucleotides, connected by bonds called phosphodiester linkages. Phosphodiester linkages. DNA and RNA are to store and pass genetic information from one generation to the next. Structure: a nucleotide consists of a sugar molecule, a phosphate group, nitrogenous base. Means they are bound together by phosphodiester linkages, and each nucleotide consists of a sugar molecule, a phosphate group, and a nitrogenous basis. Means each nucleotide consists of a sugar backbone, a phosphate group, and means this is how is nucleotide basically is, and a nitrogenous basis. Means nucleoside is another thing and nucleotide is another thing. Each nucleotide is going to contain a sugar molecule, that can be deoxyribose or ribose, and a phosphate group and nitrogenous base. This is the nucleotide structure. And in both DNA and RNA molecules, each nucleotide consists of a linear backbone that consists of a repeating sugar phosphate units. However, it's the nitrogenous bases that are responsible for storing the genetic information. Means these are the nitrogenous bases which stores the genetic information, which are going to pass down to parents to offspring. It can either in the bacteria and either in, we can say, in the human. So in bacteria also this genetic information is passed like that. DNA exists, but their DNA is not double helical structure. Their DNA is different. DNA exists predominantly in its double helix form. DNA exists predominantly, mostly in humans, in double helix form. They exist in double helix form. Double helix structure is this below structure, and the helix consists of two strands running in an anti-parallel fashion. They are held together by a variety of interaction, including hydrogen bonds. So, variety of structures, including hydrogen bonds. So, from slide number 3, we can see hydrogen bonds are this. A pairs with T, this is the hydrogen bond, and G and C, this is also the hydrogen bond. This is triple hydrogen bond, double hydrogen bond. These are the hydrogen bond. Means these are inside of the DNA. So the inside of helix consists of non-polar nitrogenous bases. So nitrogenous bases are non-polar, and the inside of the helix consists of non-polar nitrogenous bases. The exterior portion of the helix contains negatively charged phosphate group. So phosphate group exterior portion, which phosphate groups are in the DNA, phosphate groups are basically on the outside of the DNA. So they are going to contain negative charge. That's why DNA is set to negative charge at outside. So due to it is due to phosphate group there. So phosphate groups are where? At the outside of the DNA. Each strand is complementary to other one. Function of DNA. DNA molecule are used to means one strand is complementary to one another. Complementary means if you separate the DNA in the separating process, daughter cells are going to contain. Let's discuss this later. So it's the only introduction of DNA. Function of DNA. DNA molecules are used to store genetic information that can accessed by the organisms and used to build protein. Means DNA molecule stores genetic information and used to build proteins, and it is also used to build proteins. Proteins can be built by it and stores various types of information like eye character, skin character, and an iris structure. And it is written here what is the next function of the DNA. Pass down genetic information to offspring. Means DNA can pass down genetic information to offspring. Means whatever the characters of, let's say, parents have white color, like skin is white, then that parent is going to transfer its white skin color code to the offspring. And let's say woman had eye color, like it had eye color, let's say it had eye color blue, then blue is going to, if it passed from that woman, then a child will contain white color skin from man, and from woman it contains iris color. So this was the genetic information of DNA. Function of RNA. RNA molecules are used to transcribe the information stored in DNA into form that can be understood and read by cell. Means RNA molecules, let's say we have taken mRNA. mRNA basically reads the information. Transcribe means reads the information given by the cells and put it in proper manner that can be understood by the cell. Means if there is a mRNA, then some cells have given signals like you have to do this particular things. So it cannot be directly read by some other tRNA or another thing. So it could be read by mRNA. So mRNA is going to read it, and that it's going to transfer it, and mRNA is going to understand as well that type of information given by the cells. Assist in protein synthesis process. So protein synthesis process is what? It happens by the tRNA. So this is the central dogma, DNA to RNA to proteins. Composition of nucleic acids. Sugar components. Although both DNA and RNA molecules contains sugar, DNA contains a ribose sugar that lacks a hydroxyl group on the second carbon. The absence of this OH group stabilizes the structure of DNA by making it more resistant to hydrolysis. This is the deoxyribose sugar. Absence of this OH group stabilizes the structure of DNA. Means DNA has H group here at the second carbon. This is 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. Let's focus on the deoxyribose. So at the 2 prime it lacks hydroxyl group, and whereas in ribose it contains hydroxyl group at the second carbon. So what is the difference between it? Means it is the difference that if we compare from ribose sugar to deoxyribose sugar, then DNA contain a ribose sugar that lacks a hydroxyl group on the second carbon. The absence of this OH group stabilizes the structure of DNA. Means the absence of this OH group at the second carbon of DNA stabilizes the structure. Means they are not going to react with H and not going to form H2O. So this is what hydrolysis is. So this is how it is. Means it is stabilized structure. Means if we say ribose are more reactive and deoxyribose are less reactive because it lacks hydroxyl group as second carbon. So base pairing rule, I have explained it already, and this is the parent DNA double helix, and it is template DNA. Basically template DNA comes from the parent DNA and it is from the new strand, and this is new strand and this is template DNA. New strand is going to form according to template strand. If it contain A, T, G sequence, then a new strand. Firstly, if the parent has A strand there, then A here, adenine here, then new strand is going to contain T, thymine group here. That's what is the template strand and the new strand. So the copying of genetic information by DNA replication. In this process, the two strands of DNA double helix are pulled apart and each serves as a template for synthesis of new complementary DNA. Basically what happens here, these are two strands. In parental DNA double helix, these are two strands. They are going to pull apart and hydrogen bonds are going to break, and after that it's going to pull apart and each serves a template for synthesis of new complementary DNA. They are going to have complementary DNA. So this is the complementary DNA. Complementary DNA means which is complementary to each other. Like A is complementary to thymine and G is complementary to C. Guanine is complementary to C. This is what the complementary mean. So it happens like that. DNA synthesis replication, DNA, RNA synthesis transcription, nucleotides, RNA protein synthesis transcription, amino acid proteins. Means firstly, this is the central dogma. It is DNA synthesis replication. Firstly, it happened replication. Then there is a transcription process. I have already explained. mRNA works here, and it's going to read and understand by the mRNA all the information which are given by the cell, and this is the protein synthesis, which is translation. So translation is the process of protein synthesis. Means protein is going to form, which happens to tRNA. I will explain it later. What is the all process in really in description form. From DNA to protein, genetic information is read out and put to use through a two-step process. First, in transcription, segments of the DNA sequence are used to guide the synthesis of molecules of RNA. Means it is used to guide the synthesis of RNA. Means DNA sequence means it's a means transcription process. The RNA the cells guides the mRNA to work it properly. Then in segments of the DNA sequence are used to guide the synthesis of molecules of RNA. Then in translation, the RNA molecules are used to guide the synthesis of molecules of protein. Whatever I have explained previous is the same process. Basically, it guides the sequence, and then there is a formation of protein, synthesis or formation, we can say. Double-stranded DNA as information archive. Strand used as a template of direct RNA synthesis. Transcription. RNA molecules as expandable information carriers, many identical RNA transcripts. So this is the transcription process. How genetic information is broadcast for use inside the cell. Previous diagrams represent that each cell contains a fixed set of DNA molecules. Its archive of genetic information. A given segment of this DNA guides the synthesis of many identical RNA transcripts. Means most of the identical transcript can be made by this process. These are identical transcripts which serve as working copies of information stored in the archive. Many different sets of RNA molecules can be made by transcribing different parts of the cell's DNA sequences, allowing different types of cells to use the same information stored differently. Means this is all the process. This is the description of this figure, or what happens here. This is means many identical RNA transcripts. Transcription was to make which is the role of mRNA here. It is the all role of mRNA here, and it is the RNA molecule. So this is the question, that this RNA molecule, how this round upper structure is called loop, how it got formed like that, how it formed. So this is the question. So answer is here. The conformation of an RNA molecule. Previous figure shows that nucleotide pairing between different regions of same RNA polymer chain causes the molecule to adopt a distinctive shape. Being single-stranded, their backbone is flexible, so that the polymer chain can bend back on itself to allow one part of the molecule to form weaker bonds with another part of the same molecule. This occurs when segments of the sequence are locally complementary. A G segment for guanine segment, for example, will tend to associate with a CCC means cytosine segment. These types of internal association can cause an RNA chain to fold up into a specific shape. So what is written here? Few points need to be focused. Being single-stranded, their backbone is flexible, so that polymer chain can bend. So this structure was firstly single-stranded. This was not like that. It got bended like that. So this was single-stranded, like RNA was like single strand. Like RNA are here, these are single strands. These were like that. So it bended itself. Why it bended itself? Because this occurs when segments of the sequence are locally complementary. GG segment, for example, will tend to associate with CC segment. These types of internal association can cause an RNA chain to fold up into a specific shape. Means it happened due to this GCCG, this patterns. These are here the weaker bonds. These are the weaker bonds. these are the weaker bond. So they are going to bend by itself. It firstly was single stranded, then due to this CGGC bonds they are going to bend by itself, and they are going to bond each other. But these are weaker bonds. So this upper part is a loop, and this is how RNA becomes. So this is the structure of RNA molecule, which happen after bending. So that's it for now. I will explain mRNA later. What is the working of mRNA, transcription, translation, and more. It was only introduction of DNA. So I will explain more about it later. So that's all for now. Let's see in the next lecture what is in the next lecture. Next lecture can be recombinant DNA technology, which is bacterial technology of vaccines production and antibiotics production. How recombinant DNA is going to work in plasmid's drug delivery process. And next lecture can be. Stay tuned, medicos and biotics. I will be there for you. Thank you. Thanks a lot.